Hello everyone, welcome to Medix YouTube channel. This is Ghairatullah Shinwari. In today's lecture, I'll be talking about pathology. For more videos about medical lectures, please subscribe and like our channel. So, in today's lecture, I'll be talking about cell injury. Cell injury is of two types, reversible cell injury and irreversible cell injury. If you keep watching this video, you would have good knowledge about reversible cell injury at the end of this video. So let's talk about a few factors which causes cell injury. But before that, what is cell injury? A decrease or irregular function of cell and abnormal structure of the cell. This is called cell injury. So when something, a factor or anything that which we will talk about later, causes change in the function and structure of a cell, this is called cell injury. These factors can be different factors like hypoxia and ischemia, toxins, physical agents, immunologic agents, nutrition, infections like viruses and bacteria, which causes cell injury. Let's say there is increased stress. In my previous video, I have talked about adaptations, but that is in another language, which is Pashto. First of all, if there is stress, more than physiological stress in our body and our cells, our cell will try to adapt with that stress, which means it will try to cooperate with that stress. But if the stress is persistence, which means continuous, it doesn't stop, the stimulation continues and the stress increases, cell will be injured. So, as I have said, there are different factors which can lead to cell injury. Here I have drawn a normal cell, which includes our organelles like rough endoplasmic reticulum, mitochondria, nucleus, lysosome and cytoskeletals. These organelles and this cell normally functions and it has different jobs to do. For that, the cell requires ATP, which is the currency of your body. Let's say in different countries there are different currencies. In US there is USD. In Europe there is Euro. In Afghanistan we have Afghanis. Same thing is in your body. Whatever you eat, if you eat lipids, proteins, doesn't matter. Your body will convert that things to ATP, which means adenosine triphosphate. So this is the currency of your body. This is the only thing that your cell can use as an energy to function. So a cell does different jobs, like the cell membrane. Cell membrane has a few jobs, which is the important one is transport of things. So for that function, which is membrane transport, your cell needs ATP. The second thing is protein synthesis, which happens in your cell, like your ribosomes have the duty of producing or synthesizing proteins. So this organelle, which is a ribosome in here, needs ATP for that function. Phospholipid metabolism. To metabolize phospholipid, you need ATP. So again, ATP is required for all these functions. So let's say what happens when there is decreased ATP in your body. The ATP which is required for these kind of functions is available or generated from two ways, aerobic and anaerobic respiration. So there are two ways that your body converts the things that you eat to ATP. There is the one way is called aerobic respiration or oxidative phosphorylation, and the second way is called anaerobic or glycolytic oxidation. There is a major difference between these two ways. The one way, which is oxidative phosphorylation, this happens in the available when oxygen is available. So let's say oxygen is available, I have eaten something, whatever it is, a food, and it has been to my circular system. So my cells will convert this food by this way, aerobic or oxidative phosphorylation or aerobic respiration. It will convert it to ATP. But if there is oxygen available, which means if oxygen is available, one molecule of glucose is converted to 24 ATP, and this is when oxygen is available. But in the deficiency of oxygen, when oxygen is not available, this pathway is switched off and this is switched on, which means anaerobic glycolysis or glycolytic oxidation, which means this happens when oxygen is not available. But there is a difference between these two. In here, I have one molecule glucose. My body will convert this one molecule glucose to 24 ATPs. But if I don't have oxygen, my body will convert the same one molecule of glucose only to 2 ATPs, which is 22 ATP less than when the oxygen was available. So there is decreased amount of ATP in the absence of glucose, and the ATP is required where my body functions, the cells functions, as I have mentioned: membrane transport, protein synthesis, lipid synthesis, and phospholipid metabolism. All these functions requires ATP, and the ATP which I generate is from either these two ways. But let's say I have hypoxia and ischemia. Hypoxia means low oxygen of the body. When there is low oxygen available in your body, that's called hypoxia. And ischemia means low blood perfusion to an organ, which means there is low blood available to a specific organ or in your body. This is ischemia. So let's say I have hypoxia and ischemia. How will this injury occur when I have low oxygen or low blood? Let's say I have ischemia, which means low blood flow through my organ. Ischemia can happen in many ways, but especially ischemia could be obstructive ischemia, which means I have an artery right here, which I have drawn. There is obstruction in this artery. Let's say it's atherosclerosis causing this obstruction. So the blood which is trying to pass through this artery, it will face this obstruction. So in the result, I have decreased blood flow which passes through this artery. This is called obstructive ischemia, which means the blood is trying to pass this artery, and let's say this is few cells I have drawn here, and reach this area, to reach this area, but there is obstruction available, so the amount of blood which is trying to pass here, it will decrease because there is obstruction. This is called obstructive ischemia. Ischemia due to obstruction, arterial obstruction, although it could be to another reason, which means the blood has come and there is no obstruction in the artery, no atherosclerosis, but there is a problem in venous drainage. The vein which I have drawn, it cannot drain the blood and return it to the heart. So there is again decreased amount of blood, again ischemia. Normally I have written here Budd-Chiari syndrome, which means there is a syndrome in which the venous drainage of blood is decreased. So in total or in result, we have decreased blood flow through an organ, which means when there is decreased blood flow to an organ, normally oxygen is transported by blood, by hemoglobin available in the blood. So when there is decreased blood, this will mean decreased oxygen. Although decreased oxygen or hypoxia can also be of other causes, like the blood is okay, but there is problem in your lungs, which has the duty of gas exchange. You inhale oxygen and you exhale carbon dioxide. So the exchange happens in your lungs. So there is a specific mechanism for that. Or the hypoxia can be due to heart disease. Your blood is okay, the blood flow is okay, but the heart is not available to fulfill the demand of oxygen required in your body. Or it could be, the problem could be to the decreased O2 carrying capacity of the blood. There are different diseases that affects your blood, and then your blood cannot carry that oxygen. Anemias of different types. The blood is okay, the amount of blood you have, which means there is no ischemia, but there is a condition or there is another disease that your blood cannot carry oxygen. And such as other factor which can cause decreased oxygen is CO poisoning, which means carbon monoxide poisoning. So what happens here is, when your blood tries to take that amount of oxygen and transport it to one of your organs, but you have been poisoned with CO, which means carbon monoxide. You have taken too much, inhaled too much carbon monoxide, which is normally available from the gas that our waste gas that our cars and factories reproduce. So you have been poisoned with that. The thing with CO or carbon monoxide is that it binds with hemoglobin 100% more than O2. And once carbon monoxide binds or connects with the blood amount or hemoglobin, it doesn't allow oxygen to bind with the same hemoglobin. So your hemoglobin or blood is already capacitated or already taken or already connected to carbon monoxide. So this carbon monoxide rejects oxygen, and in the result you have hypoxia, because there is carbon monoxide available in your blood, and that doesn't allow the connection, or it doesn't allow oxygen to bind with RBC or hemoglobin available in RBC. So that's called carbon monoxide poisoning. Or there could be another reason, which is called methemoglobinemia. This means the iron available in your hem is normally of F2 plus. This is the state. This F2 plus iron available in hemoglobin is the foreign that we need. So this can carry blood, oxygen in the blood. So, but if we don't have that kind of iron in the blood or in the hem, let's say we have F3 plus, this is not able to transport oxygen and bend to oxygen. This normal happens or is a result of sulfur or nitrate drugs. If you take sulfur or nitrate drugs for a long time, this can cause F3 state of iron in the blood. Or people which or babies, newborn babies also have sometimes the same status. And in the result, because Fe3 plus iron cannot correctly bend with the oxygen, so this will again result in hypoxia. So in the result, as I mentioned, in the result of hypoxia, we are studying cell injury due to hypoxia now. In the result, if hypoxia is of any kind, from any factor, if it's due to lung disease, heart disease, or if it's due to ischemia, whatever the cause is, it will result in cell injury. First, reversible cell injury. So the first thing which happened in hypoxia or in when oxygen is not available, ATP is decreased in a cell. So decreased ATP, and ATP was required for what? For cellular functions. So when ATP is decreased, intracellular lactic acidosis will happen, which means lactic acid will deposit inside the cell. So when that happens, the first thing which is the result of lactic acidosis, intracellular lactic acidosis, is mitochondria will switch from aerobic glycolysis or oxidative phosphorylation to anaerobic glycolysis. It will switch the way it works for generating ATP. So in aerobic glycolysis, as I mentioned, from 1 molecule of glucose, we generated 24 molecules of ATP. But in anaerobic glycolysis, we only generate 2 ATP from the same molecule of glucose, which was 1. So this is the first thing that happens. The second thing happens is rapid depletion of glycogen. the cell with which is facing injury due to hypoxia will be low on glycogen. And the reason here is because this glycogen, when there is low ATP due to hypoxia, because mitochondria cannot make more ATPs, so the available glycogen will be converted to glucose, and it will be used in anaerobic glycolytic pathway. So why would you be low in on glycogen? The reason is simple. You generated, when there was oxygen, your cell generated 24 ATP, molecule ATP from one molecule of glucose. But in anaerobic glycolytic pathway, from one molecule of glucose, there is only 2 ATPs. So to generate the same amount of ATP which the cells require, you need more glucose. So therefore glycogen is more used. The use of glycogen is increased in this pathway. So that's why rapid depletion of glycogen, which means glycogen is used very fastly and it is low. The other thing which happens in lactic acidosis is decreased pH. In decreased pH, which is acidic pH, if the pH decreases, it goes toward acidic pH. So this will result in clumping of nuclear chromatin. The chromatin which is available in nucleus, this will clump together, as you see here in the picture. This is nuclear, this is chromatin, and I have drawn another picture here which is clumping. So it will clump together. And another thing which happens in hypoxia, in lactic intracellular acidosis, it is accumulation of lactic acid. Lactic acid would also accumulate, it will bend together. And another thing is denaturation of protein. Protein is available here in the cell everywhere. It will denature, it will lose its naturation. It will be like divided into different particles, and it will be available in the cell. That's another reason of lactic acidosis. And the third thing which happens in hypoxia is plasma membrane damage, and this is the very important step of cellular injury. So when plasma membrane is damaged, you will also see, let's say this is my plasma membrane, which is also called cell membrane, is damaged. You would have blipping of the cell, as you can see. This means blips. Let's say I would draw another one here. It will make a blip like this, as you see. And when this happens, this is where cellular injury gets worsened. So you need ATP for what? For the generation of phospholipid from fatty acid. Which fatty acid? Intracellular fatty acid. Let's say there are fatty acids available here, and your body needs ATP to generate phospholipid. Why do you need phospholipid? Your cell membrane is made of phospholipid. Bilayer phospholipid mostly. You have also other things in your cell membrane like proteins and carbs, but mostly phospholipids. So you have phospholipid from fatty acid, and it won't be generated, and you need it because to repair your cell membrane. But you don't have much ATP to do that, so the function could not be carried out. Your body cannot do that. So in the result, you have decreased phospholipid, and membrane will be damaged and blipping will occur. Why? Decreased phospholipid. Your body cannot repair that. In the result, when your membrane, there are pumps in membrane pumps. There would be dysregulation in membrane pumps, specifically in sodium-potassium ATPase and calcium ion pump. These are two important pumps in your cell membrane. So let's say moving towards cell injury and membrane damage. In the membrane damage, we will study sodium-potassium ATPase pump. When this pump fails to do its job the way it should, what happens? Normally, this pump is responsible, sodium-potassium ATPase, which I have drawn here, actually. This is sodium-potassium ATPase. The normal function of it is that it will carry out 3 molecules of sodium out and 2 molecules of potassium in to keep the balance of intracellular sodium and potassium. But when this pump fails, it cannot do its function properly. More sodium will go in the body, in the cell actually. So in the result, because the pump is not functioning properly, normally it should take 3 molecules of sodium out, but it cannot function properly, so there is more increased sodium in the cell. And when there is increased sodium in the cell, guess what happens? There is more H2O, water, because sodium has a, it observes, it functions as an observant for the water. It absorbs more water toward itself. And in the result of increased water in the cell, the cellular swelling occurs, which is the hallmark of reversible injury, cellular swelling. And this is why? Because there is more sodium. And why is there more sodium? Because you have dysfunctional sodium-potassium ATPase. So this is that. So swelling, cellular swelling is the hallmark. Another thing that happens in your body, calcium ion pump does not work properly, so there is calcium influx in your cell, and when it does not work properly, you have increased calcium. So in the result, this calcium would specially go and deposit in the mitochondria, causing the swelling of mitochondria, and it will also swell. And you would also be able to see amorphous densities, which means phospholipids inside the mitochondria, which has clumped together, these particles of phospholipid. So swelling of the mitochondria, swelling of the plasma membrane, and swelling of the ER, endoplasmic reticulum. When endoplasmic reticulum swells up, the detachment of the ribosome, which are attached to the endoplasmic reticulum, this occurs. The ribosomes, which are responsible for protein synthesis, ribosomes which are responsible for protein synthesis, they detach, so they cannot function anymore. So in the result, you have decreased protein synthesis. And why did this happen? Because of the swelling of ER. And why did ER swelled up? Increased sodium, increased water. That was the result. So in the result, you have this one also. And also microvillus detachment. In the cell membrane, you have a structure called microvillus, which are finger-like projections, and these are helpful when there is increased intracellular pressure or in the absorption. So when the pressure in the cell increases, it can, it can be helpful in that. And when there is absorption, when some molecules needs to be absorbed, it could be absorbed through microvillus. But when there is increased water and the cell swells up, the microvilli will be detached. So you would have detachment of microvilli. You would not have that anymore, that structure anymore. So cell swelling, protein synthesis is down, mitochondrial swelling, ATP is down, and that's all because of the increased amount of intracellular water. And the responsible factor for that was sodium, high amount of intracellular sodium, due to what? To the decreased ATP amount. So another thing that in the reversible cell injury you would see is myelin figures. Myelin figures are like phospholipid. phospholipids, it could be in the cell, myelin figures, or outside of the cell. It could be lying in the cell. Where these phospholipids come from, these myelin figures, which makes our myelin figures, which are myelin sheets like figures, they came from either cell membrane or cell membrane of the organelle, the intracellular organelles. So the phospholipid, which are normally available in the cell membranes of organelles and cell, they are deposit in the cell or outside of the cell, and this is also a sign of reversible cell injury. So in total, as I have shown in this picture and as I have already mentioned here on the whiteboard, in the reversible injury, things that you would see is cellular swelling, decreased amount of protein synthesis, decreased amount of ATP, myelin figures, swelling of the endoplasmic reticulum, deattachment of the proteins, deattachment of the microvilli, swelling of the mitochondria, increased sodium, intracellular sodium, and increased intracellular and intramitochondrial calcium. Up to this level, the cell injury is reversible. But if the stimulator or the factor which is causing this is stopped, if you stop, let's say, hypoxia, and you don't have hypoxia in your body anymore, which means there is no low oxygen anymore in your body, and you have good amount of oxygen, you don't have ischemia, you don't have lung disease or whatever was causing the hypoxia, your cell will convert, or your cell will return back to its normal function and normal structure. Okay, so this was reversible injury all in one video. I hope you liked the video. Please comment and subscribe our YouTube channel. For more videos and medical information, you can follow our YouTube channel, and also comment if you have required or if you need specific video or on a specific topic.